Good evening, folks, and welcome to Channel 8 Health News. I'm Scout Heath.
And I'm Collin Reimer.
Tonight, we'll be focused on scientific medical developments. Tonight, we have two guests to talk to us about the emerging new treatment, gene therapy. For our introduction, please welcome Dr. Wells and Dr. Smith. Thank you so much for joining us. Now, for the audience, could you give us a breakdown of what exactly gene therapy is?
Of course, thanks for having us. Gene therapy is on the forefront of fighting disease. Essentially, gene therapy is intended to correct mistakes in the body of DNA sequences. Your genes code for proteins. When the sequence contains a harmful error, then a protein is malformed and unable to properly perform its function.
And gene therapy works to correct that?
Absolutely. Gene therapy seeks to use viruses to carry genes to cells in the body to fix the problem. We can use gene therapy to correct genetic defects. Not only that, but we can use it to help treat cancerous diseases.
Now, it's my understanding the viruses used in gene therapy are a specific kind of virus. Care to elaborate?
That's right. Gene therapy makes use of retroviruses. Retroviruses use RNA for their genetic material and consequently use reverse transcriptase to generate DNA from their RNA in the host cells they co-opt. After that, retroviral DNA integrates itself into the genome of the host cell, and then the inserted gene can be expressed and replicated through mitosis.
That's the key here. If we can control what genes the retrovirus carries to the host cell, we can control what new genes the host cells receive and express.
It seems to me that this could take us far in the medical world, but what's holding us back?
One of the cons of gene therapy is that the retroviruses cannot be injected directly into the body of a patient. In order for gene therapy to work effectively, retroviruses must act directly on affected cells, for example, the cells of a tumor. The other method is to retrieve cells from the patient, inject those cells with the retrovirus, and then reinsert the cells back into the patient's body where the retrovirus can then affect the damaged cells.
The method of taking cells from the body and reinserting them is very difficult and a delicate task. The cells can only stay alive outside of the body for so long, and while they are outside of the body, the scientists need to make sure that the cells stay alive and are safe. Injecting the retrovirus is also important, as they must be careful not to kill or damage the cells. Finally, the cells have to be placed back inside the patient's body. This is the hardest stage because we have to make sure that the cells continue to survive once they are back inside the body, and make sure they don't cause any other problems, like changing the DNA makeup of healthy cells inside the body, aside from the pre-existing ones.
So essentially, it's extremely difficult to keep the cells viable outside and inside the body. Interesting. What else is slowing the progress with this research?
Finding a proper virus to carry the desired gene corrections is another factor. Some tested viruses are quite successful in carrying the genes and infecting cells. However, viruses can have limitations, such as only being able to infect dividing cells. The problem with this, of course, is that gene therapy often needs to be applied to cells that don't actively divide, like nerve cells.
Other viruses have been experimented with, such as HIV, but using harmful viruses means having to also remove the harmful genes within them. Removing harmful genes also reduces the efficiency of the virus's ability to infect.
Interesting. So there seems to be a lot of challenges in the way of this sort of treatment.
Unfortunately, yes, but that doesn't mean it isn't worth it. There's been plenty of treatments. In fact, the first treatment to be approved in the U.S. was T-cell gene therapy. What's more is that some success has been found, particularly with the treatment of adenosine-deaminase-deficiency, or ADA. ADA damages the immune system and causes severe combined immunodeficiency. These patients' immune systems lack the ability to fight off bacteria, viruses, and fungi. This causes the patient to be prone to repeated and persistent infections that can be seriously life-threatening. On a good note, patients with that disorder responded so positively that they no longer needed the ADA enzyme treatments previously required.
Gene therapy has also helped treat blindness caused by Leber congenital amaurosis, more easily called LCA. LCA stems from a mutation in the CRX and the IMPDH1 genes. CRX is essential in creating functioning rods and cones, while IMPDH1 is important for cell growth. When these genes are mutated in the retina cells, LCA causes continuous deterioration of the retina, but gene therapy was able to produce some improvement over several years. Not a permanent fix, for sure, but it did prevent further decay in the retina. Even similar issues like choroideremia in a trial of 9 patients resulted in 6 patients with improved sight.
That's quite a success story.
Now I'm interested in the science behind this. As we all know, our own bodies have ways of fighting back against infection. We have antibodies to protect against viruses our bodies have already encountered. What stops antibodies from reacting against viruses used for gene therapy?
That's a great question. The answer to that is antigenic drift. Antigenic drift refers to the mutations accumulating in the viral genes coding for antibody binding sites. The buildup of these DNA sequence changes can mean that while antibodies may be able to recognize and fight one strain of a disease, they might not necessarily be immune to another.
This change means we can use different strains of viruses and still be able to treat patients with previous immunity. This process is even more important because it can be applied to cancer cells. Cancer cells in the body can be taken from the body and similarly engineered to be recognized differently so that the body will act on them rather than allowing them to further grow.
Can you elaborate on the recognition of cancer cells? What exactly does that mean?
So, the immune system operates on a system of recognition so it knows which cells to attack and which ones to defend. Biomarkers are the key molecules that differentiate between normality or abnormality in a cell, and they're what's used to determine the best approach to gene therapy for a specific patient.
Biomarkers can offer a sort of prognosis of disease through the expression level of specific proteins on tumor cells, which suggests the kind of response the patient will have. Similarly, it can be used to determine the actual response after treatment, usually through blood or tissue samples taken throughout the process.
Right. So the problem with cancer cells is that they're not being recognized and the body doesn't know to attack them?
That's it. One of the major pieces of the immune system are T cells. T cells are immune cells that, unlike B cells, can be used on pathogens that operate with host cells. T cells recognize the pathogen residing in the cell when it presents biomarkers, or more specifically, MHC class I proteins on its surface. The T cell then uses toll-like receptors on its membrane to recognize the protein, and then signals for the destruction of the host cell through programmed cell death. When the toll-like receptors are not able to detect the biomarkers on the host cell, the pathogen can proliferate while the body doesn't know.
Normally, a T cell recognizes a foreign cell, releases cytokines to basically recruit more T cells, and then they all destroy the antigen. In the case of cancer, however, the recognition of the tumor cell just simply does not happen.
Right. So how do you fix that?
Well, retroviruses can be modified to carry a DNA coding for chemometric agent receptors, an artificial receptor specifically designed to detect specific cancers such as lymphoma and blood cancer, and possibly even colorectal and breast cancers. In cases where this chemometric agent receptor has been added, the modified T cells work very much like a living drug and were very effective.
Amazing. Simply amazing. As for current research goals along these lines, what can you tell us?
Well, the goal here is to develop better immune systems. Take the T cell example. Normally, cytotoxic T cells are responsible for eradicating antigens, working primarily on abnormal MHC class I proteins expressed by the antigens. In cancer, these proteins are altered but remain undetected.
The beauty of gene therapy is that because we know how the immune system functions normally, we can specifically equip it with tools to help it where it fails. Researchers can program T cells with a specific receptor to recognize the cells we want to target.
Consider it similar to a vaccine. Instead of teaching the immune system to handle a virus, we give it specific instructions to destroy cancer by putting them to the correct antigen to bind to. From here, the process resumes as normal. The T cell releases perforin and cytotoxins. The perforin creates a pore, and the cytotoxins enter through the pore and kills the cell. Retrovirus gene therapy can be used to craft a whole new immunology for patients by recruiting T cells and helping them perform their jobs.
And there you have it, folks. Retrovirus gene therapy. It's new, it's clever, and it may just be the next medical breakthrough in the age-old tug-of-war with disease. This is Channel 8 Health News. Thank you and good night.